Sex-Linked Inheritance by Jayana Tabilos, Hunter Sussman, and Kelly Draper.
When the gametes from the male and female reproductive system combine, the egg carries an X chromosome with it, and the sperm either have an X or a Y. When they combine, you can have a 50% chance of the X chromosomes matching up, producing a female, and a 50% chance that the X and Y will match up, creating a male.
Some genes work like alleles that can be linked to the sex chromosome. Here it's the X chromosome. This gene can either be dominant or recessive.
Two different examples of sex-linked diseases include hemophilia, which is the lack of blood clotting protein so that the blood does not clot properly in the body, and muscular dystrophy, which is the weakness and loss of muscle mass caused by mutations that affect the development of proteins. In this scenario, we're going to be looking at a female who has hemophilia, a recessive sex-linked disease. For her chromosomes, as of now, she has two big R's, meaning that she is not affected by the disease in any way because it's recessive. But if she were to have two little R's, that would make her affected with the disease. And if she were to have a heterozygous genotype and have one big R and one little r, she'd be unaffected by the disease, but she'd be a carrier and able to pass it on to her children.
To be affected by a sex-linked recessive disease, a male only needs one recessive allele on his X chromosome to be affected. However, if he has the dominant allele on his X chromosome, then he won't be affected by this disease. We'll be using hemophilia, a sex-linked recessive disease. Here we are crossing a female that is affected by the disease and a male who is not. As a result, 100% of their daughters will be carriers for the disease, while 100% of their sons will have the disease. Here we are crossing a female that is not affected by the disease and a male who has the disease. As a result, 100% of their daughters will be carriers for the disease, while 100% of their sons will not have the disease. Here we're crossing a female carrier for the disease and a male who is affected by the disease. As a result, 50% of their daughters will be carriers, while the other 50% will have the disease. 50% of their sons will not be affected by the disease, while 50% of their sons will be affected.
Here we observe a pedigree tree tracing hemophilia, a recessive sex-linked disease. We're trying to figure out the genotype of this female right here. To do this, we have to look at her sons, which has one affected and one who is not affected. This means that she has to be a heterozygous carrier for the disease because she has two sons that have different genotypes, and we know that the X chromosomes in her sons are from her. Here we observe another branch of the family where we're trying to figure out the genotype of this unidentified female. She has a mother who is affected with the disease and a father who isn't affected, and also a brother who's affected with the disease, meaning she has to be a carrier because she will get one affected allele from her mom for sure, and then one unaffected allele from her father.
To conclude, here is a practice problem. A female carrier for hemophilia, a sex-linked recessive disease, marries a male affected by hemophilia. What are the percentages of the affected, non-affected, and carrier children they may have? For this problem, we are crossing a female carrier for hemophilia and a male that is affected. 50% of their children will be affected by the disease. 25% of the children, or daughters, will be a carrier, and 25% of their children or sons will not be affected at all.